to the Weekly Bioanalysis, a KCAS podcast.
Hello and welcome to the 5th episode of the Weekly Bioanalysis. My name is Dominic Moreno and I'm the Senior Scientific Advisor for all things related to ligand binding assays and flow cytometry. I've been with KCAS for a little over 7 years now, but I've been in the CRO space for close to 15, and in the immunology, LBA, gene therapy, flow cytometry space for close to 25 years. I'm here with my co-host, John Perkins.
Hi, this is John Perkins, also Senior Scientific Advisor at KCAS. I've been with the company around 7 months, but prior to KCAS, I've been within Bioanalytical for 24 and a half years, so I have a wealth of experience to pull from.
So today's podcast, as always, is brought to you by KCAS, Kansas City Analytical Services. KCAS is a bioanalytical laboratory located in Kansas City, Kansas, serving the pharmaceutical and biotech industries for over 40 years now. John and I are senior scientific advisors, as I've previously mentioned, and either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS services. We're thrilled to have you listening again. As we mentioned, this is our 5th podcast, and really the exciting thing about this is this all started because we couldn't be together due to COVID. We couldn't travel as much. And now, John, it's just kind of taken off. At least for me, it's becoming part of my routine and something I'm budgeting for week over week and getting ready for this. And I'm just so excited to talk about today's topics.
Yeah, absolutely. Actually, this is the highlight of our week. One big bit of news to celebrate our 5th podcast episode. We're happy to announce that you can now follow us on Twitter. That's right. You can leave your thoughts and comments to us about the podcast by following @TheWeeklyBio on Twitter. We'll be posting our episodes and updates on that account, and we would love to hear from you.
Yeah, so every week we start our podcast by going over some news and notes, and then we'll talk about our main portion, which this week is LCMS 101 and the evolution of LCMS, our small molecule to commodity, and new challenges in LCMS and the collaborative approach between small molecule and large molecule laboratories.
Then we'll move to feedback and questions from the audience and give you then a taster of what we're likely to be talking about next week. Now it's time to move to the news and resources section of the webcast. Over to you, Dom.
Yeah, there's been quite a bit of pharmaceutical and biopharmaceutical activity in terms of funding and investment dollars and mergers and acquisitions. I think we're starting to see some companies kind of come out of dormancy now that we're well into almost 2 or 3 months or a full quarter of COVID. Unlike in 2008 when the economy crashed, this sort of dip in business has been due solely to the virus and not necessarily a lack of funding. So one of the first things that came out this week was the U.S. allocated $11 billion for COVID testing. And that's just an incredible amount of money. Certainly there's a lot of controversy around how the COVID testing is going, but $11 billion coming from the Trump administration, man, that seems like an awful lot of funding, but hopefully if it's put to good use, it will help us with testing of antibodies, and we'll talk a little bit more about some of the progression there as well as the vaccines. But John, I don't know about you, but $11 billion is a hefty amount to be infused into our economy.
It will be very interesting to see where it's applied and how it's allocated.
And then there's another company called Novavax, and they scored a $384 million coronavirus vaccine deal. I don't know how familiar you are with Novavax, but they're a European-based company, and they are throwing their hat into the coronavirus vaccine arena as well.
Yeah, I noticed that the funding comes from CEPI, who are a European organisation. They're actually based it's the Coalition for Epidemic Preparedness Innovations, based in Oslo, and the founders were actually Bill and Melinda Gates and the Wellcome Trust. I think when they work with Novavax, they started off with a small allocation of around $4 million, but there must be something promising there that they then produced a further funding of $384 million. It's interesting to see Wellcome Trust are actually further in the news where they've actually spun out an additional non-profit which they're calling Wellcome Leap, and they basically funded that with $300 million with the brief of addressing global health challenges.
Yeah, I wasn't familiar with the CEPI. Apparently it's the largest sum they've ever given. Did not know they were associated with the Bill and Linda Gates Foundation. Similarly, I saw the UK trust 300 million to LEAP. I didn't realize they were all sort of bundled together, but that really shows that there is a tremendous amount of money being poured into not just coronavirus, because the UK Wellcome Trust, that was for global health challenges. I know maybe some of that's corona, but certainly that had connotations to it that meant things other than coronavirus.
Yeah, there's plenty of other viruses, etc., that still need attention in Africa and various other places.
Another interesting investment this week was Bluebird Biosciences. And they received $200 million from BMS for their anti-B cell maturation antigen CAR T cell therapy, or their anti-BCMA. And interesting, almost simultaneously the next day, what happened, John? They got the $200 million from BMS, and then what?
They got two, and then they got refused to file notice from the FDA, which, I mean, obviously we'll see what that actually means and what the impact on their program is going to be. And then the news today was that actually another company looking at exactly the same area, Legend Biotech in China, has had launched a $100 million IPO. And then it's interesting that you've got Bluebird partnering with BMS, but actually Legend are teaming with J&J to address this area.
I think it's really a passion of mine. I love the CAR T cell therapies. I mean, I worked on what is essentially T cell therapies for a decade during the 90s. There are a lot of them starting to penetrate the market. I think COVID really put a big downturn on several of them, but I hope to see that rebound. There's two interesting other investments that are non-COVID related. And one is from Qualitative, and they got 42 million for ALS and frontotemporal dementia. So those are, the neurodegenerative space, I would imagine, has been really impacted by this, but it's really nice to see some infusion of money in that area.
It's nice to see money go there. Just it would be really also very nice to see some success in that area, because it's a very high risk area of medical research when you look at the number of compounds that just really haven't gone anywhere, sadly.
Yeah, that could be. I felt like I read something a year ago or a couple of years ago that said it is the biggest failure in science. I wonder if we had like an outbreak that caused us to be COVID-like, if we would pour more energy and resources into ALS. That's a kind of an interesting thought around how we've rallied for COVID, yet things like neurodegenerative disorders, they're still pretty, kind of toting the line as a single company and not a collaborative approach.
Yeah, but I think it's a field of trying. I mean, there's the sort of plaques associated with Alzheimer's, which seem to be an indication of it occurring, but we haven't really figured out what that indicates and what it really tells us. I think that's that's where the problem lies. I think the brain is a very complicated organ that we don't really understand yet in terms of some of these conditions.
Yeah, and you're so true, John. I mean, for the longest time, it was all A beta peptides and tau, and then it switched to like things like alpha synuclein and TIMP. And what I think we're discovering about neurodegenerative disorders is that they're kind of similar to leukemia, lymphomas. There's probably hundreds of different types of them, and that's where the challenge lies in terms of trying to treat them and cure them. But interestingly, I know the CAR T cell space is venturing into that area as well. Maybe moving on to the next round of funding, it was a company called Grail. And this is an interesting company. They received their fourth round of funding in the amount of 390 million for cancer-seeking blood tests from two large Canadian national pension boards plus two undisclosed investors. I thought this was somewhat interesting, and I think that is the sign of the times where people and investors had to press pause. They didn't lose any of their money. They still have all this money, and now they're out there kind of investing in, random things. To hear a Canadian National Pension Board investing in a biotech company seems kind of novel to me.
Absolutely.
Yeah. And then maybe switching gears, there have been a couple like downturns. One is a company called Kaiser Permanente, and they reported a $1.1 billion loss in Q1. How much do you know about Kaiser Permanente? I know they have these cool commercials.
I know very, very little about them, to be honest.
Yeah, I think they're like a health management company, and they've took a big downturn, and that made news. But really right in our wheelhouse was Charles River's Laboratory had some big losses in Q1, and that was kind of surprising. And most of that was in their animal space in terms of providing it to academic labs. Any thoughts on Charles River downturn?
Well, I was surprised by that because obviously we're seeing slowdown in clinical, and that's not Charles Rivers' normal place. We're seeing downturn in clinical, and actually a lot have a lot of discussions about non-clinical work. But I think it's one of Charles Rivers' many services. So I think other things are moving forward fine, but this one where they are obviously supplying academic labs, and that's those labs either don't have money or just aren't able to access the facilities. So the research isn't happening, so the need for animals is gone. So yeah, they're obviously it's one of those things that I don't see that business turning around anytime soon because it's majorly impacted by the current situation.
Yeah, it was on the surface, it was quite surprising seeing how I feel like we're as busy as we've ever been. And then when you hear, oh, well, it applies to academic centers, and we've both spent plenty of time in academic labs, and it makes you go, oh, yeah, that makes a lot of sense, right? Because a tremendous amount of animals are used in the academic setting. Well, why don't we move on to another topic we wanted to touch on in the news and resources, and that is how pharma and everyone's playing nice together. John, of course, the biggest news this week was from Gilead, who partnered with 5 generic companies to produce remdesivir to 127 countries. I mean, this type of partnership is like unprecedented. I mean, some of the companies in here are like Milam and Cipla. I'm not sure I know who they are, or for Zonis Laboratories, Hetero Labs, and Jubilant Life Sciences make out the 5. But it's not unheard of to have a pharmaceutical company partner with a couple generic, or at least a generic company. But to do it in 5 and to want to produce it to 127 countries, this feels a little bit altruistic to me. I don't know if they're going to be making money on it, but I thought it was a really good story this week.
Yeah, I managed to completely miss that one, but I would have to think that if they're working directly with generics, then this is it seems to indicate that they're not going to it's not going to be high priced medication, so that could be good news for everyone.
Yeah, I mean, I think some of it is to bring it. I don't know if free is the right word, but really at least at cost or very cost effectively. And then remdesivir kind of hit the news. I felt like there was some stories about it that still continues to have controversy around its efficacy and the end power behind some of the studies, and even treatment of a COVID patient at a different stage will obviously impact how effective the drug is too.
Absolutely, I think you're seeing studies where they have to look at patient population, whether they're early symptoms or more advanced symptoms, and see where it's most effective. I mean, I know we've talked about it in the past, but that was certainly the case for the Fujifilm drug that's been looked at in China and India as well. And its indication is much more for early stage with people because of the way its mechanism is to stop the replication of RNA. So it has no application at all with advanced cases.
Yeah, it's one to continue to monitor and watch. Staying on the COVID topic, specifically vaccines, Pfizer and BioNTech, they have a nice partnership. That's one we've been kind of monitoring. They're going to dose their first patient. I don't know if it already happened or certainly this month. They went ahead and ramped up several million vials of vaccines. And this is an interesting sort of thing. And it's not just specific to Pfizer and BioNTech, but anyone, all the players out there, J&Js, whoever has the GSKs, whoever has their hat in the COVID vaccine space, they're making millions of vaccines. They don't even know if they work yet.
Yeah, I mean, but the need is so acute. I mean, they have to be able to show that they have the ability to produce millions of doses if they find that it is efficacious. But it's certainly interesting to see that change in approach where we're not doing everything sequentially. There's talk of doing things much more in parallel to try and move things forward as quickly as possible. And yeah, to see the production ramp up before you've actually got clinical trials completed is certainly interesting.
Yeah, it's a head scratcher, but I guess when you kind of describe it the way you did, I guess it's making sense. But still, it just is amazing because if it's not working, then all that money just, all that investment, all that time just goes poof, right? Um, I will wrap up the news and notes now with COVID testing still dominating the news. 2 things in particular, 2 really mega, I'll call them CROs, but mega clinicians laboratories, or however you want to think of LabCorp and Quest. But they're 2 mega conglomerates that came together to team up to make COVID testing. I think this is pretty cool to see that sort of two giants who are direct competitors, in my opinion, and suddenly they're, kind of teaming up and playing nice together.
Exactly. It'd be very interesting to see what that collaboration consists of. If I can just throw one last thing in, the COVID roundup, there was actually another thing on Endpoints News. This is a summary of a study in BMJ where they did an analysis of there's 1,087 trials ongoing for or planned for coronavirus, and their verdict was that most of them are actually a waste of time. I think they've done this kind of research in the past where they've looked at they plan for studies that there isn't sufficient scientific power within them, and they've talked about this in terms of other indications. But what they're seeing is because there's so much focus on COVID and so many people moving to it quickly, it's sort of accentuated that issue within clinical trials.
Yeah, I caught a glimpse of that. It didn't make me widen my eyes and scratch my head around, wow, all of these trials are just being done and they're dosing people with various compounds and it's really for no good at all, right? Or with little efficacy.
It's this sort of running before we can walk. Again, it's like, we do need to do this stuff, but let's do it right. And they seem to be losing sight of that.
And that's some of the pressure from society in general, wanting it, as quickly as possible. But that as we say at KCAS often, science sometimes gets in the way of the expectation.
Exactly.
Going along that theme, I listened to a podcast on antibody testing this week, and I was fascinated that I believe the fifth or maybe the sixth approved antibody test. So an approved antibody test would be something that goes through the 510K process. It gets some sort of in vitro diagnostic to it. And the FDA says, hey, this is within certain limits, specificity, selectivity, this works. There are still 179 other commercially available antibody testing kits for COVID-19. And all these manufacturers have to do is validate it internally, and then they can go out and sell it as some sort of lab developed test. And this just baffles me that we've got, so many COVID-19 tests out there that really haven't gone through the process.
It just boggles my mind. I don't know how to respond to that. It's crazy. I mean, yeah, we're seeing collaboration on other things, but we need to see some unification on the antibody testing because you can't persist like this.
No, and I think it's scary because today I saw Wuhan wants to test every single person for antibodies. 11 million people they want to test. I mean, what if you just think of the end power around testing 11 million people, and if only 80% of it's accurate, that's a huge number of people you're misdiagnosing with their antibodies, right? I mean, this is crazy. I don't know. And when we do a tremendous amount of work on what is called an anti-drug antibody. And when we do that, that goes through three tiers of testing. So the COVID-19 antibody testing is just the screening component. I don't know if you know what I mean by that, John, but the screening is just, hey, you run the test and it looks like it's positive. And in our world, when we go to determine that something has immunogenicity against a drug, it goes through three tiers. So there's that screening tier, which is just the sample, all the reagents that are necessary for the given test, and you get a result. And it's above a certain threshold, it's considered positive. But then we reflex test that and we confirm it. And how we confirm that is we actually have to show some sort of percent inhibition. So it's the same test, only you're going to pre-treat the sample with the drug in our world, and you got to see like a 50% reduction in that signal. And if you don't see that, then it's not deemed positive. It's actually considered negative and a false positive. So none of that's being done with these tests. And that just kind of, for someone we got a lot of knowledge around it, it just is really troubling to see some of these COVID-19 tests.
Sure. I totally agree with you.
So why don't we move on to actually today's topics? This is going to be a lot of fun for you, John, right? So the agenda for today, look, we can start with what is bioanalysis. That's the first item.
Shall I take this off? So our thought was when we've been doing this podcast for however many weeks, we talked about bioanalysis, we've talked about Lcms, we've talked about ligand binding, but we haven't really defined what we do. And we thought it would be a good idea to maybe For this week, we'll talk about bioanalysis and LCMS as the technology. So to start, what is bioanalysis? It's bioanalysis effectively in our shop is the quantitation of drugs or biomarkers or metabolites in biological fluids, whether they come from animals or humans. We analyze samples and support our clinical trials. And what we're doing is measuring the level of drug that is in those samples so that we can then track what a drug is doing within your body after it's been administered. There's also qualitative bioanalysis, which we do things like metabolite ID, but we do less of that. There's much more interest from our perspective on the quantitative side of things.
You kind of gave a nice summary of bioanalysis more from like that small molecule perspective. And certainly you touched on the biologic side. And on the biologic side, it gets a little bit, I don't know if diverse is the right word, because metabolites can be incredibly diverse in your world. And we get things like, yeah, there's straightforward monoclonal antibodies that are therapeutics. But then we start to get into chimeric proteins. You start to get into like gene therapies and cell therapies, and then enzyme replacements. All of them kind of extend into that bioanalysis realm. And it's always within matrix. I think that's the big thing to emphasize. And when we say matrix, we mean some sort of bodily fluid.
Exactly. Thank you for the. Actually, thanks for the addition, because I always think small molecule. I need to get my head a little bit more away from that.
No, and I think I'd like to round out a little bit more. So just to touch on some of the evolution of bioanalysis. And so I think it'd be cool to take a moment and talk about, like, over the last 25 years, you've really been in that LCMS space, and you can share with us, and you'll talk a little bit about how that's evolved. And then I'd like to share from my perspective of how, up until 2001, we didn't really even have a guideline, right? There was the first hallmark paper out there that is the first one that addresses biologics, but for the longest time it was all small molecule, and only over the last, I think, maybe 5 years have we started to really separate the two. Before, they used to try to get into one bucket, and it was really challenging for the biologic side.
Absolutely. So, shall I go to LCMS?
Yeah, please.
So, okay. So LCMS, we abbreviate it because we're an industry that loves acronyms, but LCMS actually stands for liquid chromatography mass spectrometry. Chromatography is effectively separation of compounds. With a mass spectrometer, we're using that as the detector at the end of our chromatographic column. But what we're trying to do here is actually marry two incompatible techniques. So liquid chromatography actually is a high pressure technique. Mass spectrometry is a high vacuum technique. So we were trying to combine the two, and certainly years of early research, the emphasis was how can we combine these things that really don't come together? Because if you actually introduce liquid into a vacuum, you're going too much liquid into a vacuum, you're going to have rapid expansion that would then crash out your system. So early research was how can we get the eluent from a chromatographic column into a mass spectrometer, but do it at as small a volume as possible so that the mass spectrometer doesn't react to that. That was based on many years of trying various different interfaces, none of which were really rugged enough to permit what we can do nowadays. And it was actually the advent of electrospray, and also as an adjunct to that, ion spray, which was pneumatically assisted electrospray. Well, what we're doing is a mass spectrometer actually detects ions. I should have said that before. But electrospray and ion spray, I'll refer to it as electrospray as much as possible from here on in. You're actually making ions at atmospheric pressure. Then the challenge becomes how do you transport those ions that are at atmospheric pressure into the vacuum? So that's really steered the evolution of mass spectrometers since then. So going back to when I did my PhD, I was doing supercritical fluid chromatography with mass spectrometry because there was the thought that actually SFC would have less impact on the mass spec. I'm not going to go into too much detail on that, rather than LC. But electrosprays effectively put that to bed. But when we used to do overnight runs during my PhD, it was a case of I would sit with the mass spectrometer, do an injection, watch the results, do another injection. As this industry's matured, we actually find that we still do our runs overnight, but we're never in the building because we're now using autosamplers, we do repeat injections. And to go back to the quantitation, what we're actually doing, we're preparing standards and QCs in the matrix of interest, and we then read off our samples against the calibration curve. The quality control samples are there as a measure to show that the actual the calibration curve is good, and it's an independent check of the calibration curve. Another part of LCMS, we use internal standards, which are there to, because you're extracting out of a sample, and one sample can extract slightly different from another. The internal standard is there to try and compensate for that variation between samples. And as bioanalysis has evolved, we've come more and more to realize that if the best internal standard is something that is closely related to your analyte of interest. So if it's structurally similar, that can work. But if the best ones are you've replaced some hydrogens with deuterium, or some carbon 12s with carbon 13, 15 nitrogen in place of your 14 nitrogen, and that those will then behave exactly the same as your drug of interest.
There's a couple things in there that, you mentioned one of the key components of bioanalysis is what you just mentioned around internal standards and reference standards. That's a key element of bioanalysis for sure. And on the ligand binding side, it's the drug product itself. And then sometimes we'll label a reagent or something like that, and that becomes a critical reagent. And then again, that's part of the bioanalysis component of things, or even the bioanalytical component. And, John, I actually did a rotation in a laboratory that had a mass spec back in, I won't say the exact year, but it was quite some time ago now. And all I kept thinking to myself is, what in the world would you want to spend all day on that machine to get data bits out? It was massive. It made a lot of noise. And then when I found out what it was doing, I was like, why would you do that to a protein? So when you talk about the ionization and all that, you're charring things in there, aren't you?
So yeah, I'm glad you brought up the talk of proteins, because electrospray is a really interesting ionization technique in that for large molecules you can actually get multiple charges onto the molecule, and so mass spectrometers actually measure things by mass to charge. So for small molecules, because you usually just put one charge on, effectively just say it's measuring the mass. But when you're looking at proteins in the early days, where there's a lot of publications on electrospray, cytochrome C would be one of the test compounds, which has a molecular weight of around 12,000. But the mass spectrometers of the time probably had mass ranges up to about 2,000 mass. So to measure a protein at 12,000, you then need multiple charges on that to get it into the range of the mass spectrometer where you can actually see it. And that's how we can look at intact proteins, is by the multiple charging.
Yeah, to me, it's just counterintuitive, right? I want my proteins intact. I kind of want them to be in some sort of folding formation that's going to replicate how it is actually in the body and in the sample. And all you're doing when you go to put it on a mass spec is just usually sometimes you're digesting it, which comes with a lot of innate challenges to it, and then you're putting it into this thing and lighting it on fire essentially to kind of get this map of what the ions look like. To me, it's still it's powerful. I'm starting to learn more about it, but as a person who likes to look at look at proteins intact, it still kind of gets me uncomfortable, right?
There is a lot of attention in academia in terms of protein structure, etc., with mass spec, but that's more academic and certainly not where we are right now. I mean, for us, we're interested in how much do you have in this particular fluid. And in the early days, I started in bioanalysis in '94. The lab I joined had been in existence for about a year, and there were a number of other bioanalytical labs popped up about the same time. instrument of choice for people was an instrument called the SCIEX API3. It was a Perkinelmer SCIEX, and it was it was an excellent instrument in terms of it gave very good sensitivity and actually had better sensitivity than several other models that followed, both from SCIEX and from other manufacturers. But it was a unique pumping system. Where to make the vacuum within the instrument, they actually used a cryogenic head. So it was frozen to close to absolute zero. And that way, any gases within the mass spectrometer would condense onto that head, and thereby create the vacuum. One problem with it was that the cryogenic head would reach capacity, and for most instruments, it was around about 40 hours. At that point, it would then go into an automatic recycle to bring it back up to room temperature to release all the gases, and then it would initiate the pump back down to low temperature to then condense to reinitiate the vacuum again. So really the recommendation when I started was, well, you do your one run overnight, recycle during the day, and then you'll be ready to do the run the following evening. But when your time is money and you don't really want any downtime, you start to push how long you can run on that instrument. And so we got fairly good idea. We had 3 of these instruments about where they went in terms of close to 40 hours. Our best instrument ran to 48 hours. Another one was 42 hours, and I can't remember what the third one did. But we would try and stack them as much as possible with runs to get to the close to like that 45, 6, 7, 8 hour window and get all our data off before the instrument would go into recycle. Of course, if we got it wrong, the instrument would just like stop mid-run. Well, it went off into recycle mode and we'd lost data. So there was a bit of a gamble there. But it did mean that we could get much more throughput.
Sounds like it was you lost a couple times, John. Huh? And just you brought back some days of old flow cytometry and how some of the challenges we had with the instruments and how you would frequently potentially lose some data due to some of the things you're describing there in terms of run times and trying to cool lasers and stuff like that. But we'll shelve that. Maybe we got another podcast topic.
two old boys getting nostalgic about the old days. Yeah, I mean, it's ultimately the any models that followed the pumping system, main pumping system of choice became turbomolecular pumps, which are pretty much like mini jet engines that suck the gases out. But for a while, the mass specs in terms of performance couldn't match the API3 for quantitation, just in terms of sensitivity. But obviously, you've got multiple manufacturers all trying to raise the bar, and we're at the stage now where we have good instruments in terms of sensitivity. The triple quadrupole, which I'm not going to go into in terms of its mechanism, is really the workhorse for most work we do. But there's also high resolution instruments have come on the market, and they're quite routinely used for quantitation as well. Really, the big change for us in terms of bioanalytical LCMS wasn't so much in the mass spectrometers, like I say, which have incrementally improved. It was actually the introduction of 96 well technology. And what this meant was that we could process 96 samples at the same time, which really helped crank through our throughput. So where prior to that, where you'd be doing extractions in tubes, you'd maybe run about 80 samples in a run, or they'd say for example clinical samples. But with 96 what we would do 2 plates back to back, and you run 150 samples. So it then gave that possibility of analyzing samples quicker, getting data to the customer quicker, to then get a drug through to market quicker. It really that was a real game changer for bioanalytical labs. It was the adoption of 96 well technology.
I even remember that I've been at KCAS when we started to adopt that, so I recall some of that happening. I also we had API3 instruments throughout our building at one point. Now we've grown so much we had to take them out. It used to look like a Smithsonian Institute, these API3s were pretty big. And I thought that was something that was really cool that you mentioned it. It made me think of how, what a great history we have here at KCAS. I've been in the business for over 40 years. We've seen and been at the forefront of all of this. I mean, I think we even today are still beta testing instruments for people and trying to get that sense that all the heralded sensitivity and throughput. Those are all the things that not just for LCMS that applies to the LBA side.
So one thing I want to move on to is, we talk about small versus large molecule, and I think there is some perception that small molecule LCMS is a commodity, and I'd like to get on my soapbox and push back on that, because I think with the standard of instrumentation, it's easy to do LCMS. I'm not convinced it's easy. It's easy to do it well. I think you still have to have a very skilled set of hands who know the science and can get the as much as possible out of the technology to really answer questions properly. I mean, we still see a lot of challenges within small molecule LCMS. Some of it is the nature of the way drug development has gone. I mean, stability is a big challenge, particularly where a number of or there's a lot of emphasis on looking at prodrugs where you may actually you've got a compound that is designed to break down to an active moiety that actually treats the condition. So your challenge as a bioanalytical person is actually to prevent the prodrug from breaking down in samples so you can accurately measure it. We see challenges from metabolites. We see urine can be a tough matrix to deal with, because without the proteins in the matrix, you'll get non-specific binding to surfaces, and that can really make it tough to accurately measure what you're wanting to look at. So yeah, I think you really need people who know the science to really do bioanalytical LCMS well, even for small molecules. But ultimately, the most interesting new research in terms of bioanalytical is really on the measurement of large molecules.
Yeah, but before we move on to hybrid and large molecules, John, I want a couple things. The perception of small molecules being commoditized is probably more in the BEBA space, right? Bioequivalency, bioavailability studies where people are taking maybe an older drug and trying to repurpose it for something, or even it might be a generics and things of that nature. Although I think, again, I'll say perception, because I think there's quite a bit of, as you said, bad laboratories out there doing that. And, we frequently see people come to us and say, hey, can you fix this for us in the BE and BA space? But for the NCEs, the new compounds, I still think that's not commoditized. I think people really look for laboratories that know how to, drive sensitivity down, look at matrix effects, look at selectivity, understand hemolysis and lipopemic effects and things of that nature. That's all. I don't see that as commoditized, but maybe there is this perception around the BE and the BA space.
I think for some people they do, but then you need to have the conversation to say why it's not. I think you're right. With new, I do agree it's more at the generic end, but I still think that sort of mentality does seep into new chemical entities. But if that's the case, then we need to have the conversation with those people to say, okay, we see your drug, we see your structure. Here's what would concern us about the analysis of this drug. We see that particular chemical group there may be some instability associated with that. It's really that the ability to interrogate the molecular structure and almost predict where your concerns are going to come, and that's where it's the scientific background is key.
Well, building upon that, certainly things like liposomes and nanoparticles that have some sort of small molecule component to it, that is by no stretch a commoditized thing. I think that's still an area that requires highly skilled individuals. And then, of course, ADCs, I think we've talked about those antibody drug conjugates, where it's a large molecule with some sort of small molecule payload on it. I think that again requires that's not commoditized to me.
And I think maybe it's the case that as drug development is going in the direction of more complex molecules like antibody drug conjugates, maybe it's taken away from the commodity viewpoint, and because the actual entities that we're having to measure are that much more challenging.
And really that leads nicely into one of the other topics of today's agenda, and that's the challenges in LCMS and the collaborative approach. And so, I've been thinking a lot about this, not only because I wanted something to say while you were talking about LCMS, but because I think there's some truth to it. If you think of an amino affinity capture as some sort of immunoprecipitation, that requires you to load some sort of column with some sort of bead with an antibody against it and pull down. Now, on the surface, that's to me, it was something that was academic. It can be done, but it's really hard to do it in a GLP setting, is what I'm trying to drive at, because the reproducibility of that time and again the accuracy and precision of that type of pull down, it's not going to have the accuracy and precision of some sort of standard LCMS method, nor even for that matter a good solid LBA method. That's one of the challenges with doing immunoprecipitations, but it gets even further when you look at digestion of the pulled down protein. So now you've got some variability in the amino affinity capture. Then if you have to have a digestion step, enzymes in and of themselves, they are putatively supposed to cut at certain places, but every time you use an enzyme, the activity of that enzyme can vary slightly. The way the protein is going to be done now, sure, it has a sigmoidal-like curve to it, or some sort of area that 95% of it might be cut the same way, or upwards of 90%. But if you factor in the affinity capture variability, the digestion variability, we haven't even put it on an instrument yet, John. So, the expectation around how precise and accurate a hybrid mass spec method has to be lowered.
Yeah, basically the answer is you need to get an internal standard in as early as possible into the process. If you had a stable labeled version of the protein, for instance, then that's a great place to start. But I don't know how often that ever happens. For the digestion portion, once you've identified a peptide that you think is going to be a good surrogate for the protein, then we would actually commission a synthesis of a internal standard of a longer version of that peptide, with heavier isotopes. So it's a stable labeled version of a longer peptide, and that would then digest to give the peptide that we would then use to track our analyte of interest through the rest of the process.
So if I'm to understand if we were to take some sort of monoclonal antibody, and there are internal standards to pieces of that, but the internal standard is then engineered to go through the digestion process. Is that what you were saying there?
Yep, that's correct.
Wow.
So if you basically you've gone through the process of identifying your peptide that acts as the surrogate as the full antibody, but within the antibody sequence, the amino acids amino acids either side of that peptide. So what you would do is actually produce a longer version, so a peptide that includes a number of amino acids either side of that, because it should digest the same way as the antibody. But that would be labeled. So then, what you get post digestion is a heavier version of your peptide of interest.
It's like a pro peptide is what you're describing, but having worked with a lot of enzymes on proteins, they just don't behave as well as I think you're describing it. It's not as precise, right? So some of the enzymes that you use are trypsin and chymotrypsin, and they're putatively supposed to cut it X amino acids. They cut much more variable than that. And sometimes with an antibody, it won't cut all of it, because then now you're getting some of the finesse around how to do these methods is antibodies can get exhausted, right? Enzymes can get exhausted if you don't have the proper amount of enzyme, and then if you get too much, you're gonna degrade it even more. So even as you describe this process, I'm starting to feel for Dawn and how she's tackling these things. But man, and John, you haven't even begun to do the actual MS method of this yet, right? So the expectation around accuracy and precision of these methods has to be widened, in my opinion.
I think they are, for me as someone who hasn't done much of this hands on, I would actually be much more comfortable with the LCMS end of things than this piece. This is I think this is where there's much more finesse.
Yeah, well, what it's been described to me is that very piece, and that's why I think we're so poised to be the leaders in this. Or, at KCAS, we have the ability to take all of our LBA knowledge and get that upfront piece and continue to help make that a more accurate and precise project. We can do more on the upfront amino affinity capture component of it and help improve the accuracy and precision because you've got an LBA team that can help work with your LCMS team. And then obviously our years of LCMS, we're not, again, my understanding is there's less challenges when we get to that point, although there still can be. It's more on the upfront piece. And let's not forget the reason why a company or an individual is moving towards hybrid LCMS or affinity capture LCMS is because they had some sort of problem with an LBA method or a problem with an LCMS method. So it's innately already has some challenges to it.
Yeah. I mean, really, it's what we're doing up front for the hybrid LCMS is we're using the technology that you're routinely using in ligand binding, and that helps drive then our analysis. So to have the group who are very experienced on the ligand binding side of things to be able to discuss issues is very helpful. Honestly, I think we could have talked about this subject for probably another hour and a half, but it's definitely a good time to move on.
So, John, that concludes the main portion of today's podcast. Why don't we go into some feedback and questions from our listeners? I have feedback for you directly, John. I got some feedback from a friend. He had been listening to the previous 4 podcasts. This is now our 5th podcast. And he was saying how great it was. And I thought, thinking it's me who's great, right? Turns out it has nothing to do with me. Him and his family love to listen to it. You know why? Because they like to hear John Perkins talking in his Scottish accent. Unbelievable. All the time I thought I was the rock star. It's really you, John, who people are coming to hear.
I'm glad someone likes it because I can't stand my accent. Having several years of it being conditioned by my mother, who hated my pronunciation of things like butter, where I dropped the T's and things like that. So, yes, I'm not a great fan of the Glaswegian accent, but I'm stuck with it, so I'll live with it.
So, outside of the feedback from my friend, we also had Gavin ask a very, very challenging question that I think all of society wants answered. And he asks, some testing results show large amounts of people carrying antibodies to COVID-19, suggesting they were already infected. How long do these antibodies persist in someone's system, and do they prevent reinfection? So I really hate it when people say it depends, but the answer is it depends. Unfortunately, there's no single answer here. So maybe we'll start with persistence of antibodies in one's, systemic persistence of antibodies. Antibodies can last upwards of 30, 60 days circulating, but what really produces them is memory B cells. So if someone is infected with COVID and they remedy it, they should have both memory B and T cells. And so they should have what is considered somewhat of a low level of them circulating in you. And so therefore, they would be protective if they have circulating antibodies that can trigger the innate immune system. And what it does is it triggers the adaptive immune system quicker. So that's a lot of immune system talk there. But the reality is they may have a more mild version of the infection. They might not have any infection at all. Or if the other factor that comes into play here is how much of the actual virus are they getting exposed to? So if it's just a small amount, one in theory would think that's low levels of circulating memory can help prevent the infection or potentially protect that individual. But if it's a big bolus, meaning somebody that's infected coughs right in your face and you don't know about it, then maybe the central memory system can't protect you against it. So it's not an easy question to answer.
So I have a question. So if the virus is prone to mutation like flu. That's one where you're presumably your protection, although you might have those antibodies in your system, because it mutates relatively rapidly year to year, it doesn't have that long-term effect.
Correct. That's the challenge with the influenza viruses. COVID doesn't fall into that category. There are some conserved capsid proteins that are the target of vaccines. So if one, in theory, generates an antibody against that, you can be protected. But just think about, and vaccines themselves are a good example of how you protect. John, you had a hepatitis B shot, right? Well, hepatitis, you went through you had a shot, you had a boost, and then frequently they may test you and see if you have the antibody, but frequently they just do a second boost. So that's really probably the best way to answer this question is no. The first time you get exposed, you're probably not protected. But you certainly could have some benefit by having circulating antibodies. And without a doubt, that's a good thing to have. But the reality is you probably have to be exposed a few times before the immune system's primed in such a way that it prevents infection. So I hope I answered that for everybody, but there is no great answer for that.
I think you answered it fine. I think you did great. So we have one more comment to follow up on. Michael says, I noted KCAS as a bioanalytical and biomarker laboratory. You've touched a lot on bioanalytical testing and how it is related to our current COVID-19 world, but will you also be discussing biomarkers soon? That is a great question, Michael, because as we discussed topics, we thought that next week would be an ideal time to talk about biomarkers. For us for an LCMS person like myself, biomarkers are a little bit more challenging because you're measuring something that's endogenous. For Dom, that's a big part of his world, is measuring biomarkers. So we want to talk about biomarker analysis with both LCMS and with ligand binding technologies.
Awesome stuff, John. That's fantastic.
I guess that's it for today. Usually we'd be doing this on a Friday and I'd be thinking about cracking open a bottle of wine and relaxing for the leading into the weekend. But because we want our producers to have some life outside of this, where they've been doing the editing over the weekend, we're recording on a Thursday so they can edit on a Friday and they can get their family life back.
Yeah, John. Well, normally when we start on Fridays, it's much easier for us to kind of have the downslope. Now that we're doing it on Thursdays, it just means we have to go back to work now, John. It seems like when we do it on Fridays, it was all downhill from there, and I was on my way to, making some pizza.
Exactly. It was that nice lead into a nice relaxed Friday night. Yeah. Relaxed Thursday doesn't have quite the same ring about it.
But there's always Thirsty Thursday, and we don't mind accommodating our wonderful production team so that they don't have to work all weekend long.
Yep, I totally sympathise with them. So yeah, my Thirsty Thursday tonight is I've been primed by my eldest son and my other kids too. We're having a family get together over the internet doing Jack in the Box quizzes. So we'll see how that goes.
Yes.
So we're busy talking about the lead to the weekend, but for all of you, you'll get to hear this on a Monday. So I hope that you enjoy this episode, and we look forward to talking to you later in the week again.